Hi everyone, this slideshow goes with Lab 5, which is on nervous tissue. And so most of you have started looking at Chapter 11, and so you know that the nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system being the brain and the spinal cord, and the peripheral nervous system being the nerves that come out from the central nervous system. We've talked a little bit about nervous tissue and how nervous tissue has neurons and neuroglial cells, but we didn't really get into what neuroglial cells do. And there's actually a few different types of neuroglial cells. The central nervous system has its own types of neuroglia, and then in the peripheral nervous system we have another couple of types. So I'm just going to go through those in the next couple of slides. We're going to look at the central nervous system first. So the first neuroglia that we're going to look at in the central nervous system is called an oligodendrocyte. So oligo means many, dendro means branches, cyte means cell. So this is a cell with many branches. And you can see, so this is the cell body, and the branches wrap around the axons, forming something called myelin. And that myelin never fails. That was a FedEx truck. It's always a delivery when I do a video. That myelin insulates the axon and makes it so the action potentials move faster. And oligodendrocytes are the cells that get attacked with things like ALS and multiple sclerosis. So it's the myelin sheath of the central nervous system that gets attacked in most of those conditions that we're familiar with, where people's ability to control their movements diminishes because the myelin gets damaged. Next, we have the ependymal cells. The ependymal cells line all the fluid-filled cavities in the central nervous system. So they're epithelial cells, but they're just found lining the cavities that contain cerebrospinal fluid. And so they help with filtering and moving fluid. And the third neuroglia of the central nervous system are the microglial cells. And these are defensive cells. So these can act like phagocytic cells, where they eat, phagocytize bacteria, damaged cells, what have you. But they also can kind of clean up damaged neurons, that type of thing. Okay, and last but not least are the astrocytes. And the astrocytes are the most abundant neuroglial cell in the central nervous system. And from this picture, we can see that they have a supportive function of kind of holding capillaries and neurons close to one another. They seem to have many, many functions that have to do with kind of supporting the neuron itself, controlling the chemical environment that surrounds the neuron, responding to impulses. But as with all neuroglia cells, it's really hard to study them because they're on the inside of our central nervous system. Even the ones that are inside our nerves are hard to study. So we're still learning what they do. So now we have the neuroglia of the peripheral nervous system. And so those are going to include the satellite cells. And the satellite cells we see here surrounding the cell body. And their functions are similar to the astrocytes of the central nervous system. And we also have Schwann cells. And Schwann cells are similar to the oligodendrocytes. So Schwann cells form the myelin around the peripheral nervous system neurons. And we'll talk more about that as this slideshow progresses. When we look at the structure of a neuron is a really highly specialized cell. And so this end of the neuron over here is the receptive end. It has the dendrites and the cell body. And the dendrites and the cell body both receive information from other cells, whether it's a receptor or another neuron. And that information if enough information comes in that that neuron wants to send its own message, it's going to send that message starting right here at the what's called the axon hillock, and it's going to travel down the axon through something called saltatory conduction. So saltatory conduction is the hopping of an action potential kind of to these nodes, these myelin nodes or nodes of Ranvier, and it's much, much faster than it would be if the impulse had to travel all the way down the axon. Let me just go through the cell body structures a little bit. So we are familiar with the nucleus, and inside the nucleus is the nucleolus. There's endoplasmic reticulum in a neuron, but the endoplasmic reticulum has two special names. You can either call it Nissl bodies or chromatophilic substance. So those are the names that we use for the rough endoplasmic reticulum in neurons. And the last structure that I need to talk about is down here, these axon terminals. So that at the very, very end, the axon branches to these axon terminals. And the axon terminals are going to synapse with another neuron or a muscle or a gland. So here is a slide of an ox spinal cord, and we can see this is a motor neuron cell body. We can see all of its branches, and we can see the glial cells that are around it. Over here is the axon hillock leading to the axon. I'm not going to ask you to be able to identify that. I think that that's a challenging thing to identify. But the body and the nucleus and the glial cells are pretty reasonable for me to ask about. I mentioned that the axon terminals are where the neuron synapses with another cell. And so we're just going to look at the axon terminal a little bit. So here's a cartoon of the axon terminal. And so the action potential is going to travel down the axon. And once it reaches here, it's going to trigger the opening of voltage-gated calcium channels. And calcium is going to rush into the axon terminal. And when calcium rushes into the axon terminal, it tells these synaptic vesicles that hold neurotransmitters to release the neurotransmitter. So we see the vesicles making their way to the cell membrane and releasing neurotransmitter through exocytosis. So, there's many, many different neurotransmitters. Probably the most common one that we talk about is acetylcholine. And so, we can imagine this being acetylcholine in the synaptic cleft. That's the space between the two neurons. And that neurotransmitter is going to eventually bump into, this is a chemically gated channel. And when the neurotransmitter attaches to the chemically gated channel that has the right active site for it, it's going to open the channel and allow for ions to move either in or out of the cell. Okay. If enough movement happens so that the membrane potential changes to reaches threshold, this cell is going to send an action potential down its cell membrane and send that signal to the next neuron or send that signal to the next muscle or gland. Here just covers how the myelin sheath in the peripheral nervous system forms. So that Schwann cell is going to wrap around the axon. And it's going to keep wrapping around that axon until you get multiple layers of cell membrane. And that's actually the myelin sheath. And on the very outside, we have cytoplasm. We have just all the cytoplasm and organelles of that Schwann cell. So this is the myelin sheath. This is the nucleus of the Schwann cell. And we can see this in a cross-section. This is a cross-section where we can see the axon, we can see the myelin sheath, and then we can see the cytoplasm on the outside. The next slide is a longitudinal section of a nerve. So a nerve isn't just one axon. A nerve is a bundle of axons. And we're going to be seeing more about the structure of a nerve shortly. But so what we're seeing here is a bundle of myelinid axons. So in the center, you can kind of see these lines, and those lines in the center of each of these kind of columns are the axons. And so here is a Schwann cell. And right here is that myelin sheath gap, or that node of Ranvier. You can call it either thing, that's fine. Okay, and so there's really not a ton of space at these nodes, right? The signal is going to go boom, boom, boom. It just hops from one node to the next, and it makes it much faster to send a signal down the axon. Here we have some different types of neurons. There are multipolar neurons, which tend to be motor neurons, and multipolar neurons have many branches going in many directions. Okay, we have bipolar neurons, and those bipolar neurons have two branches, one in each direction. Here we see the major branch here and the major branch here. And these tend to be special sensory cells, so olfactory, retinal cells. They are in the special senses. And then we have unipolar neurons, and unipolar neurons have one process and the cell body is kind of off to the side. Unipolar neurons are sensory neurons. They tend to be sensory neurons. So information comes in here, information goes out here, and these are special senses. So I want to go to the structure of a nerve. So I've mentioned that a nerve is a bundle of myelinated axons, but this is going to get into the bundle of this is going to get into the details a little bit more. So here's an axon and its myelin sheath, and wrapped around that axon and myelin sheath is tissue called the endoneurium. A bunch of axons with their myelin sheath and endoneurium bundled together forms a fascicle, and a fascicle has connective tissue wrapped around it called perineurium. A group of fascicles and their associated blood vessels have connective tissue wrapped around it, and it's called epineurium. So epi is the covering, peri is within, and peri is between, and endo is within. That's the way to remember that. And over here we have a cross section of a nerve, so we can see those axons and the myelin sheath. We can see the perineurium because this is a fascicle. It's a fascicle at the edge of the nerve, so this is the epineurium. And let's see, we can see the endoneurium, so that's the connective tissue that's around the myelin. And so that's the structures that we want to talk about. I've had a few people ask questions about collaterals, and I'll be honest, there's really no place to label collaterals on what we have. And even if we met in lab, there wouldn't be a place to label the collaterals. What they want you to know, and there's actually, I'm over here, but there's not really a collateral. Oh, actually there is, right here. This branch over here off the axon, that's a collateral. Okay, so you just need to know that a collateral is a branch off the axon. And I could use an illustration like this, but I can't use any of the models that I posted or pictures that I posted other than this one. Okay, I hope this helps you in your studies of nervous tissue, and I hope you join me later on this week for dissecting the sheep brain. Take care.